possibly both, they have the retina the other way around, the one that does make sense. So they have the light sensitive cells at the front, then a layer of neurons, then a layer of blood vessels to supply oxygen and nutrients. That makes sense, right? So they're very likely independently evolved to have the same function, to be able to see your surroundings. Yeah? And there's obviously lots of molecules involved. Some of those molecules might actually be the same because they're even older. Okay. Yes, so that's convergent evolution. Yeah. When we're talking about homology, what kind of evolution do we have? It's not convergent. It's the other one.
Divergent.
Divergent. Right? You start from one point, the common ancestor, and you get two things that are still basically the same, but in appearance, in details, are different. Okay. Right. That was one slide. Right. So, how does Go do that? How does Go annotate these functions? And the first letter in Go is for gene. That's easy. You got that. So we have it here. The second is ontology. Who knows what an ontology is?
It's like giving terms to your label, right?
So giving terms to your label. This is dictionary. An ontology is also a dictionary, but it's more than that. So who knows the difference between an ontology and a dictionary?
The study of being.
Sorry?
The study of being.
Study of being.
Yeah, with the philosophical
So it might be
definition.
the literal Greek translation of the word, but it has a different meaning here. Like homology has a specific meaning in biology. Okay, so an ontology is a structured vocabulary or dictionary, right? And I'll explain that in a minute. So it defines these things. We already have them, and so this is so if you say we have a thing which could be a car or a locomotive, or a car could be a big car or an engine. This is still a bit of a dictionary. But the structure comes in the thing where you actually define in your dictionary that a locomotive is a type of thing, and a car is also a type of thing. And then you have a big car, which is a type of car, and you have an engine, which is part of the car. So there is, in addition to the terms, which have defined meanings, you have the relation between the terms. Okay? And then you have two types. So what are the two types of relations that you see here? Has, yeah, I hear somebody say has. Yeah, so one of them is labeled. It says has. So a car has an engine has a cylinder. You could also do it the other way around. You could say part of, where you say an engine is part of a car, or it can be part of a car. And the cylinder can be part of an engine. Yeah? And there's different meanings to the word cylinder. So you have different terms that could all be labeled cylinder, but then they're not in the same relation to engine. They might have, some cylinders have no relation to engine. Okay? So that's one of the reasons that you want to have an ontology. It's to disambiguate. Right? So one word can mean different things depending on the context. Yeah, like homology in English has a different word than it has in English. I thought I saw a question, sort of half. It's already gone. Okay. I hope it's good. Okay, so has or part of, if you invert it, that's one. What's the other one? It is, yeah, or is a, or is a type of, right? So a person is a type of big car. Big car is a type of car, a car is a type of thing. Yeah? And now, what else do you see on this slide? Can be. Sorry, what?
A thing could be an object, it doesn't have to be an object.
Oh, right, yes. But so actually there are two ontologies here. This is ontology A, defined by people from the A community. This is ontology B, from people from the B community. And they've been describing a very similar thing, but in a very similar way. And one of the nice things you can do once you have an ontology, or once you have two ontologies describing similar things, you can actually try to connect them. It's called ontology mapping. And we don't go into that in detail now, but it's good to know that you can actually do this. Where you say, well, a thing is usually an object. Yeah? Horsepower is always horsepower. A locomotive is sort of similar to a train, but it could also sometimes mean something slightly different. A car is almost always an locomotive. Yes? And then you can start connecting. So if you think about machine readable use of data, and one database is using this ontology, the other one is this one, you only have to do the mapping of the ontologies once, and you can start mapping data from the one database to the other. Yeah? So it doesn't mean that ontologies solve everything, because they also create problems, because you have to maintain them, and they are like all models and all data, they are an abstraction, because there's things that you can easily say with an ontology, and there's things that if they're not defined in the ontology, you can't say. So you either have to invent a new term, or you use a combination of terms to try and say something that you couldn't. It's like what I have a lot when I'm lecturing, you're trying to find the word that you're looking for because it's exactly the right one. And you can't find it, so you have to invent something, a description of 5 or 15 words. Yeah, so you have a similar thing here. But they do make a lot of things right. So this is general ontologies, and now we have the gene ontology. This is a structured vocabulary. And so this is what it looks like. And here, this Go uses is a. So it starts with biological process. That's one of the roots. That was the word I couldn't find here. So these are the 3 roots. So there's biological processes. There is molecular function and cellular location. Those are the 3 roots. So there's 2 types of biological. I don't know, possibly more, but on the slide there are 2. So biological process could be physiological or cellular, and then there's also processes that are both. They have a separate annotation, but they're also annotated here. And then there is cell division, which is a cellular physiological process. Then there's cytokinesis, which is a type of cell division. And then there's cytokinesis after meiosis, which is a type of cytokinesis, which is also linked to meiosis. Right? So this is how this works. So this looks like a tree, but it's not. Why is it not a tree? Because it has one root, so it could be a tree because trees have one root. But it's not a tree because, because in a tree branches don't merge. They might look like they merge sometimes when they're grown together, but they don't. Yeah? A tree only branches out, that never branches back in. You see here that this branches back in, this one comes back in here. So in computer science lingo, this is called a DAG, a directed acyclic graph. Because you can't go in circles, that wouldn't make sense, because then you have a circular definition of something. Yeah, that's not useful. But you can branch out and back in. Okay. And there's more relationships, but this is enough to know for now. So this is how Go is used to annotate functions. So if you have this particular function attached to a gene, then you actually have all of these terms annotated onto that gene as part of the function, the annotation of this biological process. Okay, so these are the 3. I already mentioned them. We went into them in detail. So the molecular function is what the molecule does, the gene product, and the process is what it's involved in. What does it do its function for? Yeah? It's like it's an enzyme. So it's an enzyme that does this particular reaction. That's the molecular function. If it's involved in the citric acid cycle, that's the biological process. Yeah? Okay. Some more examples, but I'll just show. So this is metabolism as a biological process. This is molecular function for chromatin binding, for example. And then you see here, oh yes, it's good to point out. So here you see, so you can take a gene-centered view, where you look up the annotations for a particular gene. But what you do here is you do the other way around. You look at the annotation structure, and you look at which genes are annotated on this branch of the tree. Right? And then you see a couple of genes here. I see almost three colors. You can probably see three colors. And one of them is yeast, the other one is Drosophila, and the third one is mouse. Or rat. Was it rat? Was it rat or mouse? Rat. Mouse. Okay. Yeah, okay, makes sense, right? Any question about this? Good. We're done with Go. All right, yes. So the Go database is actually the database that holds the ontology. That's not useful in itself. Well, it's very useful in itself, but it's not very exciting. Because what you actually want to have, and what people mostly mean when they say Go, they mean the gene annotations made with the Go ontology. So if I'm going to look something up in the Go database, and you usually mean I'm going to look at the annotations for a gene. Not I'm going to browse through the ontology structure of the Go database itself. Okay. Annotation, yeah, so this Go term. So there's a Go database and there's a Go term database for annotation. The other thing that's important for Go is the evidence. So how do we know that this is true? Or why do we think this is probably true? Yeah? Or why do we think this might be correct? So there's different levels of confidence attached to certain facts. Because each annotation you can see it as a fact. Right. And with the evidence codes there's usually a reference paper. Not all. So these are the different evidence codes. So it can be experimental, directly from the lab, let's say. It could be a statement in a paper, author statement, with the reference there, or somebody remembers that it was in a paper, but they can't find the paper, and they think, well, I should put it in anyway, right? So it was in a paper, but we don't know which one. Or the curator thinks, oh, we know that it's true. But, why do we don't know why. He forgot. Right? So then you just put in, there's no evidence, but it's in there. Right? And then the other one, which is interesting, because that's where we come in as mathematicians usually, is IEA, inferred from electronic annotation. Yeah? That's derived inferences. So it could be, the most common one is by homology, which usually means you have a gene, you blast it, you find 10 genes that have a Go annotation, and they're similar, you just paste that onto your new gene. And then you hope that the function is actually conserved in that homology, because the function doesn't have to be conserved. If you have two homologous genes, if the mutations are in the wrong place, or just the right place, depending on how you look at it, it might actually have a different function. It might not bind this enzyme, this ligand anymore, or it might not be able to function to do this kind of chemistry, but it does something completely different, or very different in some cases. Yeah.
What you touched upon now, isn't that the problem of these kind of databases? That over time they grow less reliable.
No, because it's annotated.
Yeah, but
You can see in the database.
You place that annotation on your new protein file without actually validating if that is true for those proteins.
No, but you annotate that this is an electronic annotation. It is an electronic inference. At some point, somebody might publish a paper that say, well, we actually measured what this gene does, and it's doing something very different. So then you update this annotation. That's why also the Go database, or the annotation database, they have a small army of annotators working on this on a daily basis.
So it's different when it's actually validated. Then it gets a different annotation?
Yeah, so the annotation can be updated because it's wrong, or it can be updated because you get better evidence. Yeah, or it can be demoted because the paper that was originally there to support the fact was found to be contradictory, and you say, oh well, this is not reliable anymore. You could leave it in with no evidence, for example, to say, well, it could still be true, but we don't know anymore if it's true. Or yeah, so that's I think the main task of these curators. There's two things they do. Right? They make sure that there's no nonsense going into the database, but they also try to keep it up to date with developments of knowledge as much as possible. Which is, of course, quite a huge task.
After you finish this master's,
Good question. I would hope yes. Yeah, I think we do something. I don't know of, the reason I hesitate is because I don't know of anybody who actually did that. But,
So there isn't like a specific master who
not that I know of, no. Okay. Right. We need to speed up a bit because we have no time left and 2 databases to do. Fortunately, we've actually covered, with this one long slide, we've actually covered a lot of that already. Because Go does function. Yeah, that's quite clear now. PFAM does sequence. Yeah, there's also a bit of structure in that, but it's mainly sequence. And this is just a recap. So it's a collection of protein families, and they're represented by in a couple of ways. The main thing, the one that you usually see, is the multiple sequence alignment. So some of these families are quite large. There could be thousands, a few thousand proteins in there. So you have an alignment of a few thousand sequences, showing what this family looks like. You could look for conserved residues, you could look for variable regions, you could look for all sorts of interesting things. One of the complications is, and it's actually also slightly true for Go, is that certainly in eukaryotes, proteins tend to be complex in the sense that they have multiple domains. So a domain is like a, I'm going to actually show you. So, this is what a multi-domain protein looks like. How many domains do you see? This is the structure of a protein. Who's not seen a structure of a protein before this slide? So this is, so you can, so this is colored by a rainbow color from terminus to terminus. So you can start at red over here somewhere. Over here, and that's the C-terminus. So it starts with blue over here, that's the N-terminus. It goes all the way here, back, and then it goes here, and then it goes here, and then it ends up there. Yeah? So, how many domains do you see? 6. Wow. Like 1, 2, 3 in the back, and then 1 in front, and then something like that? Yeah, well, there's clearly, if you look at the structure, there's clearly, this is one domain, right? This is separate from the rest. So there's at least 2. If you look in scope, where this is a structure database, this is how it's annotated in domain boundaries. There's 3 domains here. Okay? So you start here with the red domain, then you get the yellow domain first, part of it, then it goes into the red, then it goes into the blue, then it comes back to the red domain, and then the sequence ends in the yellow domain. So they are close in structure, but in sequence they're separate parts. They form one domain. The whole story on how that can happen, but that's not for now. But that's just to support the thing. So typically, each of these domains have an associated function. One could be the catalytic site, where the chemical activity is. Another one could be DNA binding, or it binds to a protein complex, or it's the regulatory domain, or it could be a couple of enzymes that are involved in the same process and they're put together in one gene. Yeah? Lots of different things. So you find that it's in Go as well, because one gene can actually have multiple functional annotations. Probably each of them is associated with one domain. In PFAM they make this explicit, because if you're looking at the sequence, it's much easier to say, oh, well, I see 3 different parts here. Right? And what's one of the ways in which evolution invents new functionality is by combining functional elements, domains from different ancestral proteins. Okay, so PFAM groups proteins by domain, so it's actually not a database of protein families, it's a database of domain families. So it means that one protein, if it has multiple domains, can actually belong to multiple different families. Yes? Same is true for SCOP. Okay. And it could look like this. Yeah, so you have different functions associated with different. So there's also functional annotations in PFAM. Does it make sense? If you have all the data to actually also annotate functions and things like that. I'm not sure if they are actually linked together. It would be good if they were. Maybe one of you can check and tell, because I didn't check recently. Okay. They're using the Markov models to, you'll get a lot of those in the next course, other than sequence analysis. I'm not going to explain those now, but they are a way to, they're a sensitive way to find similarity between sequences. And these hidden Markov models, they are based on a small set of confidently aligned sequences. And why do we know they're confidently aligned? Because there's a curator, a human person who actually looked at the sequence alignment and said, this looks good. Yeah? And then you make a Markov model out of it, and with that one you search for more sequences that are similar to the ones you put in your seed alignment. This builds your protein family database. Okay, so this is what it looks like on the website. This is kind of our domains. It should be. Oh, too fast. And there's another important thing about PFAM is that there is an A and B part. A is just the curated seed alignments and the Markov models, and the B family is all the other additional sequences that are aligned there. So you can think of this as being the more primary side of the data, and this is more the derived data. So this is quite confident. It's human curated. We expect few errors there. There will be some errors. We expect few of them. This, you could say, is like the automatic annotation in Go. These are basically predictions of homology, or yeah, you could say they're predictions of homology based on the information in the single line. Right. There's also a structure in that. I don't have time to go into that. Now, SCOP is about structures, and actually basically the same thing you can say about sequences. You can say about structure, although structure comparison is a little bit more complicated than sequence comparison. You can basically do the same thing. So it's also split into domains, as I already mentioned. I already showed this one, and it's a hierarchical. It's hierarchically organized. Maybe there's some hierarchy in PFAM as well. I'm sorry, I missed that. We really have to stop now. I want to get to the end of it. Like in Go, but much simpler. So there's 4 classes, and then the folds. There's multiple folds per class, and then there's multiple superfamilies per fold, and then there's multiple families per superfamily. So you may want to look into this, because homology information is stronger at the family level than at the higher level. If the proteins are in the same class, it doesn't mean they're very similar. But if they're in the same family, then they are very similar, and they're more likely to be. This actually should really be homologous proteins only. They're going to be the same family. Okay. This is another view of the tree. These are the 4 classes. This is the same thing again. And this I don't have time for. Right. So, there was a little bit about data, more about data. So this is the take-home messages for today. Are there any other questions about the lecture? No? Okay. Thanks for being here. Thanks for your very interesting questions. And I'll see you in a few minutes in practical.